Madam, you have seen all the places in Hyderabad.
No, sir, only the tourist places.
No.
Charminar, Golconda.
There is one place called Tank Bund.
Best place I have visited is that Indian Institute of
Business Management.
National Institute of Animal Biotechnology, sir.
Okay, NIAB.
NIAB.
It is located in Madhapur or Mayapur somewhere.
Madhapuram.
Madhapuram. But actually, I have requested Mohanty sir to give 4 names from the same university. So he has given.
Actually, I think all have
Yours is the last one.
joined also. Recently, within 1 year, few assistant professors they joined. In extension, medicine, LPM.
If they are interested, just let me know, madam. Just
okay, sir.
So I will speak to them.
Okay, okay.
Basically, this is a major issue, madam. HS is a major problem in the field. But unfortunately, nobody knows about it. Nobody means even the field vets, they are not much aware about this topic, HS. Vamshi sir, what is your opinion? You can unmute your microphone, sir.
Yes, sir. True, sir. It is one of the major problems, sir, and more often it is being underdiagnosed, and it is a silent killer because these organisms are normally present as commensals in the nasal mucosa. Whenever the animals are being exposed to stress condition, especially transportation.
Stress is the main predisposing reason.
Yes, and overcrowding. And overcrowding due to other environmental changes, these organisms will produce different syndromes, primarily septicemia, pneumonia, and mastitis, and it is mostly underdiagnosed, sir. Even though farmers are giving HS vaccine, but the vaccine doesn't contains active components of organisms. That is the main problem, sir.
Not only, sir, cattle, sheep, goat, other wild animals are getting infected with this HS, Pasteurella multocida.
Okay. Wild animals, huh? Okay.
Wild animals, sir. Mainly, sir, deer during the transportation from one zoo to another zoo, new place. That stress also induces this
stress is the only reason, madam. Whatever the species.
Definitely, sir, stress is the only factor for Pasteurella multocida infection.
But the definition of stress is very vast, madam. See, just people are thinking that stress means by walking itself is comes under stress. But it is not just walking. Under nutrition, handling at the time of vaccination, handling at the time of deworming, transportation.
Yes.
And as Vamshi sir said, overcrowding, particularly intensive, is a major issue.
Environmental factors plays main role as
Madam, now it is 6. I will start the program. Participants. Vamshi sir, okay?
Okay, sir. Okay, sir.
Okay. మీ బ్యాక్గ్రౌండ్లో సౌండ్ వస్తుందా సార్?
లేదు సార్.
ఫ్యాన్ సౌండా లేకపోతే ఇంకేదన్నా? ఓకే. లేదు మీరు పెట్టండి, మీరు ఆన్లో పెట్టండి, ఆన్లో పెట్టుకోండి. Good evening all the participants. I am Dr. Ashok Kumar Valpadasu, Deputy Director in Veterinary and Animal Webinar Series organized by Sathyazerograzing, exclusively on sheep and goat sector. And today's webinar is organized jointly along with Dynamic Agro Machines. And these webinars are organized under the heading of CLIPS, Continuing Learning Intensive Farming Skills. Topic of today's webinar is Pasteurellosis in sheep and goats. Generally, we call it as hemorrhagic septicemia HS. Speaker is Dr. Ashabari Samal, Professor. I welcome today's guest of honor, Dr. S.M. Vamshi Krishna, and today's speaker, Dr. Ashabari Samal. Welcome both of you. I welcome all the 4 permanent invitees of these webinars, Dr. Arun Kumar Tomar, President, Indian Society for Sheep and Goat Production and Utilization, and he is the present Director of CHWRA also. Dr. Manish Kumar Charlie, Director, ICR, CARG Mathura, Dr. Chanda Nimbkar, Director, Animal Husband Division, Nimbkar Agricultural Research Institute, Paltan, Pune, Dr. G.V. Ramanjanelu, Executive Director, Center for Sustainable Agriculture. Respected participants, the main aim of organizing these webinars is promoting sheep and goat production in India and promoting zero grazing. In general, we call zero grazing as stall feeding or intensive farming. And the main objective is capacity building of field veterinarians. Capacity building of field veterinarians by dissemination. For this, we have been conducting live webinars since last 3.5 years, every Sunday at 6 p.m. And so far, we have conducted 178 webinars. And today's webinar is the 179th webinar. Our other activities for strengthening the field veterinarians are, we have a WhatsApp channel wherein daily updates are posted in that WhatsApp channel. And we have one YouTube channel also with the name of Sathyazerograzing, and it is open for all, which are exclusively made on sheep and goat sector. And one can find all the recordings of previous webinars also in this YouTube channel. And this kind of activity is first of its kind in India. Our knowledge partner, Dynamic Agro Machines. With regard to today's speaker, Madam Dr. Asha Bari, presently working as Assistant Professor at College of Veterinary Science and Animal Husbandry at Department of Veterinary Microbiology, OUAT, Bhubaneswar. She has completed her BVS and H in 2001 from Orissa University of Agriculture and Technology and completed her MVSc in Bacteriology and Virology in 2008 from same university and completed her PhD in 2012 in Veterinary Bacteriology from IVRI. Her MVSc thesis was molecular characterization of lactobacillus species isolated from ducks, and PhD thesis evaluation of diagnostic potential of recombinant MPT63 protein in bovine TB. Her area of specialization in PhD, isolation, identification of bacteria, PCR, cloning and sequencing, gene expression, SDS page, Western blotting and ELISA. Regarding to her job experience in the relevant field, she worked as veterinary assistant surgeon and worked as research officer at Odisha Biological Products Institute. And presently, she is working as assistant professor since 2012 at Department of Veterinary Microbiology. Regarding the scientific publications, research articles 14, popular articles 16, conferences, symposia, seminars attended 10, and professional membership in different scientific societies 7. Here is the list of training she has undergone and faculty development training at ICR NARM Hyderabad, faculty development training at IIM Sambalpur, Odisha, frontier approaches in diagnosis and control of animal viral disease with quality assurance and quality control of veterinary biologicals, safety and potency testing of veterinary biologicals at IVRI, bioinformatic tools for genome analysis IVRI Izatnagar, and experimental training methods EEI Hyderabad. Regarding fellowships and awards, she has received University Merit Scholarship during her MVSc program between 2006 and 8. And she is recipient of University Gold Medal in MVSc in 2008. And she has been awarded Indian Council of Agricultural Research NET in the year 2010. And she has received Best Poster Presentation Award in 2012. Regarding professional membership, she is a registered member of Odisha Veterinary Council, registered member of Veterinary Council of India, and life member of Indian Association of Veterinary Microbiology and Immunology, life member of Indian Association for Advancement of Veterinary Research, life member of Laboratory Animal Science Association of India, life member of Veterinary Public Health Specialist, and a life member of Indian Poultry Science Association. This is in brief about today's speaker, madam. Now I request today's guest of honor, Dr. S. Vamshi Krishna, working as assistant professor, and he is head of the Department of Veterinary Microbiology also. He is working at College of Veterinary Science, Mamnoor, Warangal in Telangana State, in the university PVNR Telangana Veterinary University. Now I request you, sir, to kindly address the participants.
Thank you very much, sir. Greetings and good evening to all the participants and esteemed veterinarians who are attending this webinar. I would like to express my sincere thanks to Ashok Kumar Valapudasu sir for inviting me as guest of honor for today's webinar. And I am glad to see my senior Asha Bari madam, who is my senior in PhD in IVRA. And I am grateful for you, sir, for extending your valuable service to our profession by conducting these kind of webinars, which are really useful to the field veterinarians and even to faculty. I came to know about your WhatsApp group and your YouTube channel before just elections, and I have seen your many of the videos and your topics. Whatever information you are providing is very brief and very unique, which all the important points you are highlighting and presenting in a short. I truly appreciate your efforts, sir. And coming to today's topic, you have selected the right person, right topic, and during the right period, sir. Because pasteurellosis, which is one of the most important bacterial disease, silently affecting many sheep farms. It is widely prevalent across India and in many developed countries also, sir. And this is the disease which is facilitating other infectious because stress is the most important for aggravating this pasteurellosis in sheep and goat, and it is associated with many different kinds of syndromes, especially septicemic pasteurellosis. It is one of the most important disease, and most often this disease is being underdiagnosed at the feed level, and whatever the symptoms we can see only for 2 3 days, and most of the animals will die, especially young ones, lambs and kids. They often die because of fulminating pneumonia, which is associated with other bacterial and viral diseases also. So once these animals are exposed to stress, these organisms which are present as commensals in the nasal mucosa, they will proliferate and they will allow other pathogenic bacteria and viruses which are causing high mortality in farms. So I am glad that Ashabari madam will go in depth about this topic and will help the many veterinarians across the state who are present in this webinar. Thank you very much, sir, for giving me this opportunity to be a part of this webinar. Thank you, Ananda.
Thank you, sir. Thank you for accepting the invitation and participating as guest. Now I request today's speaker, madam, to kindly start her presentation.
Good evening, all the esteemed participants present in this webinar. Respected Ashok sir, first of all, I would like to thank you for inviting me to present this important topic, that is on pasteurellosis in sheep and goat. I would like to thank Dr. Vamsi for joining the presentation.
Yes, madam. First click on share screen button, then.
So today's topic
Make
is
it full screen, madam. Make it full screen, madam. On the top, go to slideshow and yes, please continue. Please continue, madam.
So today's webinar topic is pasteurellosis in sheep and goat. Pasteurellosis is one of the important bacterial disease in sheep and goat. Generally, we are concerning about pasteurellosis in cattle. But these Pasteurellosis infection generally occur in sheep and goat. Not only cattle, sheep, goat, also so many ungulates like other wild animals like bison, deer, and many wild animals, they are affected due to Pasteurellosis because this particular disease that occur only due to stress, and these are normal the commensals of the our upper respiratory tract and the lower respiratory tract, also sometimes in the nasal tract. So they are generally the normal common sense, but whenever there is any stress, any kind of stress, whether that is the environment or whatever, handling stress, overcrowding, these stress factor, all these stress factors, they elevate the infection of Pasteurellosis in the respiratory tract. So this Pasteurellosis, this is generally the pneumonia, respiratory disease, pneumonia of the respiratory system. Mainly the pneumonia, respiratory pneumonia are due to many factors. Mainly the bacterial causes are there, viral causes are there, maybe due to parasitic infection or maybe due to fungal infection. So pneumonia is one of the factor which is causing respiratory distress and stress to the animal. So respiratory diseases such as pneumonia are one of the main concern in the livestock industry, and economic losses due to pneumonia is prevalent in small ruminants, particularly in the case of sheep and goat. There is a very economic loss which generally we ignore or it got unnoticed. So pneumonia is nothing but the inflammation of the pulmonary parenchyma, which is accompanied by inflammation of the pleura and bronchioles. Devastating, this is very, pneumonia is very much devastating in young animals like kids and lambs, and they commonly cause of the high morbidity and mortality in kids. The causes of pneumonia are often classified into primary type or secondary type. Primarily infection may be due to bacterial infection, viral infection, or any other infection, and mostly this respiratory infection occur due to the secondary bacterial infections. Secondary bacterial infections are more commonly due to the Pasteurella infection, which are generally they are normal present in the respiratory tract. They are the normal commensals which are always present in the respiratory tract, but during stress they get elevated. So the predisposing factors are transportation stress, starvation, winning, then strenuous exercises, exposure to bad weather during hot and humid condition of the climate, then drought or poorly ventilated ones with accumulated feces and urine. All these predisposing factors lead to the infection of pneumonia or respiratory tract infections, and more particularly Pasteurella infection. There are many causes of the pneumonia, maybe bacterial, viral, parasitic, mycotic, or mechanical causes of pneumonia. The bacterial causes of pneumonia are mostly due to the Pasteurella infection. Among the Pasteurella, the important one we know is Pasteurella. Important bacteria is Pasteurella hemolytica or Mannheimia hemolytica. The next group of Pasteurella is Pasteurella trilogy, or nowadays this is popularly known as Behalogy. Then other causes of bacterial infections are also mycoplasma, which causes contagious bovine pleuropneumonia or contagious caprine pleuropneumonia in bovine and caprine population. Then Pseudomonas infection, along with that Corynebacterium infection, Staphylococci, Streptococcus, Salmonella, Klebsiella pneumoniae, all these are the bacterial causes of pneumonia. The viral causes of pneumonia are the IVR virus, then parainfluenza, bovine respiratory syncytial virus, along with Medivisna, Jaxiate, and pox viruses. Then, among the parasitic diseases, Dictyochilus, Cystochilus, Protostrongylus, Neostrongylus, these also cause pneumonia in the lungs. And mycotic cause it is the Aspergillus fumigatus, which causes Brooder's pneumonia. The mechanical causes of pneumonia, which is due to the faulty medication or faulty dosing, which is commonly known as the drenching pneumonia, drenching medication. Now coming to the pasteurellosis infection in sheep and goat. Pasteurellosis, this is commonly refers to bacterial infections of the lower respiratory tract. The causes are Pasteurella hemolytica or Mannheimia hemolytica, Pasteurella multocida, and Weber's trilogy. These are the primary pathogen for pasteurellosis infection. Mannheimia hemolytica is the most commonly isolated bacterium, followed by Weber's trilogy and Pasteurella multocida in sheep and goat. The disease generally occur in pneumonic form in all ages and in septicemic form in very young lambs. So, Pasteurella multocida that causes pneumonic pasteurellosis and arthritis, and Mannheimia hemolytica that causes pasteurellosis along with mastitis, and Bibestinate trilogy or Pasteurella trilogy biotype B that causes septicemic form of the Pasteurella. Then hemorrhagic septicemia, HS, commonly known as HS. HS is the second most reported disease in India during the last 2 decades. This is as per the National Expert System, and this is the cause of maximum number of deaths reported in livestock in the country. Even after the vaccination of routine vaccination, regular vaccination of HS, since still there is occurrence of HS in our country, and HS is a fatal systematic disease of ungulates causing great losses mostly in Asiatic country and Africa, and this is characterized by rapid courses of infection, that is edematous swelling of the throat, then edematous swelling in pharyngeal and brisket region also. There is swollen and hemorrhagic lymph nodes and the presence of subserous petechial hemorrhage on the visceral organs. Then shipping fever. Shipping fever, this is the ovine and caprine bacterial pneumonia. The pneumonia which occur in the ovine species and caprine species, that is commonly known as the shipping fever. Then this is caused by Mannheimia hemolytica. This is less the mainly the causative agent is Mannheimia hemolytica rather than Pasteurella multocida. The clinical disease is usually most severe in young lambs and kids. If at the early stage of the infection or in acute stage, then the affected lambs or kids, they appear dull and they become inactive or powerless before the death in a matter of within few hours, and the animal will be dull and inactive and powerless. Sick animals are often unobserved until the final stages of disease. They are unnoticed. The symptom without the diagnosis, they got died. In the older animals, the signs of the respiratory disease are observed, like there will be dyspnea, slight frothing in the mouth, there will be cough, nasal discharge. These signs are bring about when the herd or flock is moved and the affected animals fall behind the rest. So when the affected animal will behind the, they will be lagged or they will remain behind the rest of the active stock, then only the infection is noticed. That time there is high rise of temperature, maybe up to 100 Fahrenheit, and there is depression and anorexia also. This accompany with the respiratory signs. Then Pasteurella multocida. This negative gram negative non-motile bipolar, they doesn't grow on the McConkey agar and they are indole positive. These are serologically, they are distinct type. They are basing on the capsular structure. They are serologically 5 distinct type. They are A, B, D, E and F. Rimmer and basing on the capsular structure of the Pasteurella multocida, they are 5 distinct type and 16 LPS type, lipopolysaccharide type, that is number 1 to 16. In Asian serotype, B2 is common, means the capsular group B and serotype 2 is common. B2 is common in Asian serotype and in African serotype E2 is, that is as per the B and 6E, this is common for Pasteurella infection. Then many proteins are found. Among the secretory proteins, about more than secretory proteins in the Pasteurella infection, and those important genes are PLP, B gene, LPP, then outer movement protein OMPA, OMP, H, and other fumarate proteins are there which are secreted, and they can be used as your vaccine candidates, preparation of the vaccine candidate. Then pasteurellosis caused by Mannheimia hemolytica of sheep and goat are called the hemorrhagic septicemia. They are widespread in distribution. Generally, they occur anorexia, recumbency, and there will be sudden death. So, in acute form, there will be sudden death in the lambs and kids. Then, chronically affected animal, they have the reduced ability to thrive and survive further infection. Significant impact on the productivity efficacy in sheep. The pneumonic form is caused by Mannheimia hemolytica, and the systemic disease is caused by Weberstenia trialogy. So, pneumonic form and the septicemic form. The septicemic form is very acute form where there is sudden death of the animal without showing any symptom. Then sheep, Pasteurella hemolytica or Mannheimia hemolytica and Weberstenia trialogy, they share common serotyping system. They comprising of total serotypes, and 90% of the all isolates are serotypable. So the isolates which are um, subjected to the agglutination test, those are mostly 90% they are serotypable, and the serotyping is done basing on the capsular antigen or the LPS antigen. Then Mannheimia hemolytica biotype A strain are responsible for the pneumonic pasteurellosis in sheep and goat, and trialogy, Weberstenia trialogy biotype T that cause systemic disease in the young 6 to 10 months old lambs. Mostly the young lambs are affected by Weberstenia trialogy or biotype T strain. Then the etiology is Mannheimia hemolytica type A2, A6, A13. These are most common for pasteurellosis in sheep and goat. Mannheimia hemolytica A2 is routinely reported mastitis sheep. So the A2 is very important for pasteurellosis in sheep goat along with the respiratory pneumonic form. Also from the mastitis cases, this Weberstenia trialogy, that is T3, T4, T10 and T5, they have been most often associated with the systemic and septicemic form of the pasteurellosis, mostly in lambs. These serotypes have been regrouped to Weberstenia trialogy biotype 2, and a new biotype 4 has been added to this Weberstenia trialogy biotype 2 groups. The presence of multiple Pasteurella spectral population in check. Multiple population of Pasteurella means Pasteurella hemolytica, Pasteurella multocida, then Weberstenia trialogy, all they inhabit in the respiratory tract. So the multiple population or the presence of many number of species, they keep the bacterial population in check by inhibiting the multiplication of the other bacterial species. So there is competition between their growth in the respiratory tract. Biotypes and biochemical characters for the identification of this particular bacteria. Mannheimia hemolytica and Weberstenia trialogy are identical in their morphology. Commonly, as you know, these are the gram-negative organism. These are aerobic cocci or cocobacilli. Then beta hemolytic type of growth in blood agar. They are non-motile, non-spore forming, and encapsulated. Why the name biotype A? Because Mannheimia hemolytica that ferment arabinose. So the name A, and Weberstenia trialogy or Pasteurella trialogy that ferment or utilize trialose. So the name biotype T. Then the colonies of the Mannheimia hemolytica are small grays with narrow zone of hemolysis in the blood agar. After 24 hours of incubation, there will be narrow zone of hemolysis for Mannheimia hemolytica, but for Weberstenia trialogy, there will be larger growth, larger colony with brownish center. In the blood agar, there will be typical growth for trialogy and Mannheimia hemolytica. Then The cultural character for Pasteurella multocida is in blood agar there will be liquid type of growth or slimy growth with smooth watery colonies. There will be moist, liquid and slimy growth. Then in the nutrient broth, you will find granular deposit at the bottom of the test tube in nutrient broth. And in McConkey agar, McConkey lactose agar plate, there will be colorless colony generally as they are non-lactose fermenter. Along with those, other biochemical tests include they are positive for indole, then epidemiology for the Pasteurella infection. As we know, Pasteurella species are the commensals of the upper respiratory tract of some sheep and goat. The very young labs are highly susceptible to the disease than the adult sheep. However, the disease occur in all age group of the sheep and goat, but young ones are highly susceptible for Pasteurella infection. The mode of infection body, mainly the capsule. Capsule is the primary necrotic toxin. There are the important virulence factor. Capsule that inhibit the phagocytosis and intracellular killing of the organism. So the cells can easily escape the post-immune system and they can replicate inside the body. Then the fimbria, which is present on the cell wall, that enhance the colonization and the proliferation of the bacteria in the upper respiratory tracts. So fimbria helps is toxic to the endothelium and it can alter the leukocyte function that act as the site. It destroys the leukocytes and platelets. Then dermal necrotoxin that help in the progressive infection, respiratory tract infection. Tox A gene, one gene is present in the toxin, that is Tox A, responsible for production of the toxin, which is present in conserved region of the Pasteurella multocida chromosome. In the clinical symptoms like definitely there is high rise of temperature, anorexia, dyspnea with head and neck strain. Then there will be open mouth breathing will be difficult. Then unilateral discharge, serous type of nasal discharge, abdominal breathing with harsh, and typical symptom is crackles long sound upon the auscultation. There will be characteristic crackle sound. Then also acute type of mastitis seen in case of sheep. Then there is lacrimation, nasal discharge, drop in milk production and anorexia. This is the serous nasal discharge in goats. Then PM lesion are very typical to Pasteurella infection. Like there is hemorrhages in the subcutaneous tissues and congestion of the lungs, then froths in the trachea. There will be lymph glands will be swollen and edematous, then necrotizing, ulcerative pharyngitis. The lungs will be cyanotic and intensely congested. Then trachea is also hemorrhagic and filled with froths. There is widespread bacterial emboli. Then severe congestion of the mucosa of the stomach and intestine. In the lungs, characteristics fibrinous bronchopneumonia, but without effusion into the pleural cavity. At necropsy, in the lungs, there is consistent findings non-collapsing lungs. The lungs will be non-collapsing with varying degree of consolidation covered by fibrin and cranioventral consolidation with red-gray discoloration. They are very far gray discoloration of the lung. And other PM findings are in case of peracute hemorrhagic septicemia, there will be enlargement of the lungs. Lungs are enlarged with congestion and there will be pinpoint hemorrhages on the surface of the lungs. In case of acute HS also, there will be enlargement of lungs along with congestion and pinpoint hemorrhages. In case of acute HS, if you see the trachea, there is congestion and froths are filled in the tracheal lumen. On the left side, you can see the froth-filled lumen of the trachea. Then pinpoint ecchymotic hemorrhages on the epicardium and endocardium. There are hemorrhages on the epicardium and hemocardium in the endocardium. Then in case of chronic HS, congestion and diffuse consolidation of the lungs. There will be typical marbling of the lungs in case of the chronic septicemia. In pathological lesions, and that is suppurative bronchopneumonia, there will be hemorrhage, congestion, and intestinal wall thickening. Then there is severe exudation, cellular proliferation, and infiltration of the mononuclear cells in lungs. Proliferation of the fibrous cells were also seen in chronic cases. The important is our diagnosis. Diagnosis mainly depend upon the history and the clinical symptoms. Sometimes the history of this transportation or stress is the pillar to the basic approach for diagnosis of the disease. The samples to be collected in case of live animals, blood, the nasal